If a neurosurgeon presses a silver medical probe directly into the exposed surface of a conscious patient's brain, the patient feels absolutely nothing. The organ responsible for processing every single pain signal in the human body is completely numb to physical pain itself. To detect tissue damage and feel physical discomfort, the body relies on specialized sensory nerve endings called nociceptors. This animated diagram of a brain tissue cross-section, the parenchyma, illustrates the paradox. It is entirely devoid of nociceptors. When stimulated, it produces a flatline electrical signal. There is zero sensory input. So if the brain tissue itself is completely incapable of physically aching, what exactly is hurting when you experience a severe headache? The sensation of a headache is a somatic illusion. The pain is strictly an extracerebral event, an issue occurring entirely outside the brain that is masquerading as internal pain. To locate the actual source of the pain, we have to shift our focus outward from the brain tissue to the biological structures immediately surrounding and protecting it. This is the extracerebral zone. It encompasses the layered membranes, blood vessels, nerve networks, and muscles that envelop the skull. Headaches occur exclusively when mechanical pressure or chemical imbalances irritate these specific surrounding structures. To understand exactly how a headache functions, we need to build a structural map of these pain-sensitive zones, layer by layer. Identifying the exact anatomical origin of the irritation is the only definitive way clinicians can distinguish a harmless tension headache from a life-threatening pathology. This 3D wireframe model establishes our baseline, a completely empty cranial cavity housing unfeeling, semi-transparent brain matter. The first anatomical layer we add is the meninges, specifically the thick, outermost protective membrane known as the dura mater, glowing here as it wraps the brain. Second is the cranial vascularity. We thread a dense network of arteries and veins over the meninges and throughout the interior of the skull. Third, we add the cranial and cervical nerves that physically interface with these membranes and vessels. Finally, we overlay the external muscularity, the thick muscle groups of the neck and the thin muscles of the scalp, along with the facial sinus cavities. Unlike the brain tissue underneath, every single one of these specific structural layers contains an exceptionally high density of nociceptors. These 4 anatomical zones are the exclusive physical real estate where all head pain originates. To understand how the brain misinterprets signals from all these different locations, we have to look at the primary sensory conduit for the head and face, the trigeminal nerve, or cranial nerve 5. This animated diagram maps the physical branching of the trigeminal nerve. Notice how this massive complex spreads across the jaw, throughout the face, and deep into the protective meninges. This creates a structural bottleneck. Whether nociceptors fire because of an issue in the neck muscles, the blood vessels, or the dura mater, the electrical data all funnels into this single nerve network. Those converged signals are then routed directly down the thickest part of the trigeminal pathway and straight into the brainstem. When the brain processes this sudden influx of data from a shared neural highway, it struggles to pinpoint the exact peripheral source along the branch. This results in referred pain. A physical irritation that originates in the neck muscles or behind the eyes is perceived by your conscious mind as a generalized, deep ache inside the head. Despite the wide variety of anatomical triggers, the illusion of internal head pain relies entirely on the routing architecture of the trigeminal highway. Clinicians divide headaches into 2 main categories. The first is primary headaches, defined as conditions where the pain mechanism itself is the disorder. There is no underlying disease. The tension-type headache is the most common primary headache presentation in humans. It is initiated by environmental triggers, such as psychological stress, acute physical fatigue, and poor anatomical posture. This cross-section demonstrates the immediate physiological response to those triggers. The muscle fibers of the scalp and neck involuntarily contract, squeezing tightly together. As the muscles sustain this contraction, they physically compress the small blood vessels running through them. This causes ischemia, a localized reduction in blood flow that rapidly depletes oxygen in the surrounding tissue. This ischemic state directly irritates the nociceptors embedded within the muscle tissue, forcing them to fire distress signals. Because of where these muscles are located, this localized ischemia translates into the classic clinical symptom, a dull, tight, band-like pressure wrapping entirely around the skull. Tension headaches represent a mechanical and ischemic crisis of the external muscular layer. If we move our focus away from the external musculature and look inward at the cranial blood vessels and their intersecting nerves, we find a different trigger. Migraines are distinctly neurovascular events, operating through physiological mechanisms that have nothing to do with basic muscular tension. A migraine begins with abnormal electrical brain activity that directly alters the local chemical environment. A sudden fluctuation in neurotransmitters, specifically serotonin, destabilizes the behavior of the cranial blood vessels. This close-up of a cranial blood vessel shows the mechanical consequence of that chemical shift: rapid vasodilation. The blood vessel violently expands, physically stretching the nerve fibers coiled around its exterior wall. Nociceptors in the vessel walls are highly sensitive to physical stretching. Every time a pulse of blood pushes through the expanded vessel, those stretched nerve fibers fire intense pain signals along the trigeminal nerve. That rhythmic pumping of blood through dilated, hypersensitive vessels is what causes the classic throbbing or pulsating pain of a migraine. This neurochemical cascade also triggers secondary systemic symptoms, routinely causing severe nausea and extreme sensitivity to light. A migraine is a chemically induced mechanical trauma inflicted on the cranial vascular network. Secondary headaches represent a separate diagnostic category. These events indicate the presence of a destructive underlying pathology. Acute structural pathologies, such as physical head trauma or the growth of an intracranial tumor, fall into this category. Infectious pathologies also trigger secondary headaches. This ranges from highly localized sinusitis to severe, life-threatening conditions like meningitis. This dual split-screen diagram compares the pathophysiology of these two infections. On one side, severe inflammation causes a swollen sinus cavity to exert massive outward pressure. On the other, infected meninges swell, pressing forcefully against the skull and the brain surface, crushing the local nociceptors. Because these represent acute damage, clinicians look for major red flags. The first is the thunderclap headache, a sudden maximum intensity pain that hits instantly, strongly indicating a severe vascular event or hemorrhage. The second red flag is systemic, a severe headache accompanied by a high fever and strict neck stiffness. These are the hallmark indicators of a meningeal infection. The third red flag is any headache that progressively worsens in the days following physical head trauma. Secondary headaches signal acute structural damage or invasive infection within the extracerebral layers. The human brain contains no nociceptors and is entirely incapable of feeling pain. The true anatomical generators of all head pain are the surrounding structures: the meninges, the vascular network, the cranial nerves, and the external musculature. These structures are activated by three primary mechanistic triggers: localized muscular tension, chemically induced vascular dilation, and infectious inflammation. Regardless of their origin, these pain signals converge upon and are transmitted directly into the brain via the trigeminal nerve. Mastering this precise spatial and physiological model is the required bedrock for accurately diagnosing and eventually treating the entire spectrum of human headaches.